Echeagaray, and I am a PhD candidate here at San Diego State University. I work in Dr. Mark Sussman's lab, and today I will be talking to you about cardiac cellular senescence. Heart disease is the leading cause of death in the US, and scientists continue to study the heart with the objective of understanding cardiac pathologies and develop therapeutic strategies. Statistically, heart disease incidence increases as we age. And while there are many factors that can influence whether we're going to suffer heart disease or not, like genetic makeup and personal lifestyle, assessing the impact of age will allow us to understand the context in which hearts typically begin to deteriorate. In our lab, one of the lines of research includes assessing the changes that take place in the senescent heart, not only in the contractile unit of the heart, which is the cardiomyocyte, this brick-looking cell that is mostly known for its role in cardiac contractility and function, but also these cells that live in the cardiac interstitium, which make up around 70% of the cells in the myocardium. These cells are constitute a heterogeneous group and is made up of endothelial cells, fibroblasts, and even immune cells. These cells communicate with each other. They interact with one another harmoniously in a way that it allows for our heart to remain functional and healthy. So now that we know more about the cardiac constitution, how do we study cellular senescence? Well, in our lab, we target a very specific step in the intracellular flow of biological information that goes from DNA to RNA all the way to protein. As you may know, the information and instructions for life are in the cell's nucleus in DNA sequences. These instructions are copied into an RNA transcript that is subsequently shuttled into the cytoplasm, where the cellular machinery will take those instructions and generate a functional protein. In our lab, through single-cell transcriptional technology, we target this intermediate step and capture a messenger RNA to derive biological insights on what's happening in the heart. Because while these cells, all these cells will have the same set of instructions in the DNA, the transcriptional profile will be distinct and unique to each one of these cell types according to their function, and most importantly, for the purposes of our research, their age. So what have we learned using this methodology? Observation of old and younger hearts have shown us that the balance and heterogeneity that exist in our heart in all different cell types shift as we age. Particularly immune cells start getting a more inflammatory profile as we age. And overall, the non-myocyte population start getting less and less efficient at going through the cell cycle, which is critical for cardiac maintenance and homeostasis. Even the process of flow of information becomes less efficient as the RNA transport pathway gets affected as we age. These findings are important because they hold the potential to delay the process of cardiac senescence. It allows us to trace these changes at a single cell level and resolution, and increases our ability to generate potential preventive and therapeutic approaches to each and every one of these changes. Thank you so much for your attention.